So far we've only talked about really basic dominant and recessive. If something's dominant, you see it. If it's recessive, you do not see it. But that's not actually how it always is, but it's really easy to learn the beginnings that way. So let's now talk about what happens if it's more complicated than just simple dominant and recessive. So in the P's that Mendel saw, that's another reason why he was really lucky, was everything was just dominant or recessive. But he kind of picked it that way. But that's why his results came out so clear. So even though there may be some complexities with dominance and recessiveness, the principles of independent assortment and segregation always still apply. So don't forget about those. So this is when it may be a little bit different than we talked about. So if alleles are not completely dominant or recessive, we're going to see different outcomes. We'll talk about what that means. When a gene has more than 2 alleles, we're going to see different outcomes. And when a gene produces multiple phenotypes, we're going to see different outcomes. And this is when those 3 examples are when we're only looking at 1 gene. So when we're done with these, we're going to talk about what happens if you're looking at 2 genes. But for right now, looking at a single gene. So just think like flower color, and that's it. So there's different degrees of dominance. So complete dominance is when the dominant gene completely covers the recessive gene. So in the case of purple and white flowers, you either have purple flowers or you have white flowers. If you're purple, you do not see the white. And if you're white and you get a purple gene, your whiteness is gone. Okay? So that is complete dominance. So the dominant homozygote and the heterozygote are identical. But there's a couple of ways where that varies a little bit. For example, incomplete dominance. This is when one is not totally dominant over the other. So the middle one gets a mixture of the 2. So is there a picture here? For example, here in our parent generation, our P generation, we have a big C, or C big R, and a C big W. So red and white, homozygous for both of them, dominant and recessive. And then we cross them, and with 1 copy of each allele, the guy's pink. He's not one or the other like we would have expected him to. So that is incomplete dominant. One is not completely dominant over the other. Okay? Then, as we go on to the F2 generation, after we self-fertilize from the F1 generation, we see the same ratio we saw from regular dominance. We have 1 that's CR, 1 that's CW, and 2 that are CR. Okay, so we get the same ratios, even though we get the same genotypic ratios, even though the phenotypic ratio is a little bit different. So that is incomplete dominance. And then there's codominance, which is where the 2 genes, they both show up. The 2 phenotypes are both there. So the 2 dominant alleles affect the phenotype in separate, distinguishable ways. So, doesn't give you an example for that one. So the example I'll give you is blood type. Okay, we're not going to talk so much about the exact specificities of blood type, but basically you can be A, B, or O, or AB, right? And so that means you get the allele for A, or you get the allele for B, or you don't get any allele, which makes you O, but you can be codominance where you see both of them. For example, if you get an A from your mom and a B from your dad, you're going to be AB. And that is, it's not a mixture of the 2, it is distinguishably both. Okay, so that's codominance. Both phenotypes are apparent. Both of them, okay? So for example, in this flower color, if this were codominance, all of the flowers that are heterozygous would be red and white. Not a mixture of the 2, but they would be red and white. Okay, so this description is on page 272, and this is really important that you read it, okay? Because this will make everything make so much more sense to you. So when we're talking about dominant and recessive, it's not that a dominant gene takes away from a recessive gene or anything like that. The alleles don't actually interact that way. The important thing to remember is that these genes are all consequences of the DNA, which is came to life through proteins. So all of these genes that we're talking about are proteins, okay? So if you have a copy of the dominant big P for purple and a copy of the little p for white, it's not that both of those genes are still making proteins, okay? So if you're big P, you're making purple proteins, and if you're little p, you're probably not making any proteins. So what's going to happen is you're going to show up purple because some purple is being made. Okay, so if you have two purple genes, you're obviously going to be purple because you have two genes making purple proteins. If you're heterozygous, you have one big P, one little p, you still have purple genes being made. And then if you're homozygous recessive, you're two little p's, you're not making any purple proteins, so you're going to be white. Okay, so everything comes back to the fact that we're making proteins from genes. So keep that in the back of your mind when we're talking about this. So in Tay-Sachs disease, this is an example of it's a heritable disorder where there's an enzyme that, if you have it, is dysfunctional. So remember, enzymes make reactions happen. They help pathways of our metabolism go. If your enzyme is dysfunctional, a lot of lipids build up in your brain, and that's bad. It takes away from brain function, and it's fatal. Okay? So at the organismal level, the allele is recessive. Okay? So if you're looking at the whole person, the allele is recessive. You have to have two copies of the Tay-Sachs allele to have Tay-Sachs. That's what that means. When looking at the whole person, a whole person does not have Tay-Sachs disease unless they have two copies of that allele for this enzyme that doesn't work. Okay? Because for your body to build up lipids in your brains, just one, you need to have two copies of the enzyme that doesn't work. Okay? If you have one copy that does work, it's going to break down the lipids and you're going to be okay. But if you have two copies that do not work, all the lipids are going to build up in your brain and you have Tay-Sachs. Okay? So at the biochemical level, so looking at the actual enzyme, the phenotype is incompletely dominant. That means that the one that works is working, the one that does not work is not working. The one that works is not covering up the one that doesn't work. It's still not working. But the one that does work is doing so much work at breaking down the enzymes or breaking down the lipids that you're okay. Okay? And then at the molecular level, the alleles are codominant. They're both made. They're both there. Okay? So that's kind of confusing. Try and wrap your brain around that. So a lot of people think that just because an allele is dominant means that it's the more common one, but that's not really true. It just means that it is seen instead of the recessive allele. So there's a gene for polydactyly, which means having extra fingers and toes, and this gene is dominant, but it's still really rare. So only one out of about every 400 babies in the United States is born with an extra finger or a toe, even though that gene is really dominant. And actually there's some islands that have really segregated populations, and I have no idea where they are off the top of my head, but since it's a dominant gene and since they only have each other to share genes with, it's very, very, very common in these island populations to have extra fingers and toes. So think about that one. Okay, so we're done talking about single alleles. We talked about regular dominance, incomplete dominance, codominance, and now we're going to talk about what happens if it's more than one allele. Most genes are not dominant and recessive. They usually have a spectrum of them. For example, blood type is one of these. We have A, B, or O. And so being A, B, or O can give you a large number of blood types. So that's multiple allele. It's not just two alleles, it's as many as there are. And then, here's blood type. So, so the alleles are known as this. It's I. So it's IA, IB, or little I. And so this will code for the carbohydrate A, the carbohydrate B, or no carbohydrate. So if you're A, if you only have A or IA or I, then you're going to only have A carbohydrates on you. If you only have IB or I, then you only have B carbohydrates on you. If you have IA and IB, you're going to have both codominance. They are both very evenly there. And if you have little I, I know you can't see this, but you have no carbohydrates and you are group O. So remember that. That might be on your quiz. And then there's something else where a lot of genes affect more than one phenotype. So that would be like one gene affecting more than just your eye color, but also affecting your hair color or something like that. For example, if you have sickle cell, that's one gene. It's one nucleotide mutation that leads to sickle cell, but there's a lot more than just one symptom, right? Same thing with cystic fibrosis. It's not just one thing that gets affected. You have a ton of symptoms that come from one gene. That's what pleiotropy means. Okay, so now we were talking about one gene at a time. This gives us different types of dominance, multiple alleles, pleiotropy, but now we're going to look at what happens when different genes interact with each other, which happens all of the time. So a lot of traits are determined by 2 or more genes. One is epistasis. And epistasis is really cool. It's one of my favorite ones. And it's when a gene at one locus, remember, one spot on some chromosome, affects the expression of another gene. So for example, you guys all know Labrador retrievers, I'm assuming, labs, different dogs. And you know there's golden labs, chocolate labs, and black labs. And the reason for this is because of epistasis. So actually, labs, any lab, there's only 2 pigment colors that they make. They make black or brown pigment. Big B for black, little b for brown. Black is dominant, brown is recessive. All labs, even yellow labs, all labs make those 2 pigments, one of the 2. There is no pigment for yellow fur. But there is another gene which decides whether or not that pigment is going to be put into the fur color. So dogs can make the pigment, but they don't have the protein that puts the pigment into the fur. So if that's the case, then they're going to be yellow because they don't have any pigment in their fur. And this is called epistasis. So we see a very, very distinct outcome for epistatic genes. So what happens is these guys down here are all little e, which means they do not put any pigment into their fur, which is why they're golden. They have no pigment in their fur. So this guy right here would be making brown pigment. This guy would be making black pigment. These 3 would be. But they can't put the pigment into their fur, so they're yellow instead. Now, that's why chocolate labs are so rare. They're only 3 out of 16 because they have to have the recessive pigment gene and the dominant protein to put the pigment into the fur gene. So, epistasis, you need to know that. And then polygenic inheritance is when there's just a lot of genes affecting something. And it's usually like along some spectrum. So for example, human skin color is a spectrum. You know that there's people who are really, really, really white, like me, and people who have really dark skin. And it's not just one or the other. There's skin colors all the way in between. That's because it's affected by a lot of genes. Your book talks about there being 3 different genes, A, B, and C, and your skin color being entirely dependent on how many dominant copies you have. So for example, if you're dominant for all 3, if you have 1 copy of the dominant allele for all 3, A, B, and C, you're going to be the darkest you can be. But then if you only have 5 dominants instead of 6, you're going to be a little bit lighter. If you have 4, you're going to be this color, etc., etc. So the number of dominant alleles you get directly affects the color of your skin. And then, obviously, I'm sure you could tell this to me, it's not just the genes that you have, it's the environment as well. For example, sometimes in the summer, I'm a little bit less pale than I am right now because I go outside. So that's not dependent on my genes, that's an environmental factor. It's not that my genes are telling my skin to become darker, it's that I'm taking in sunlight, which causes chemical changes in the pigment in my skin. Okay, so the environment can also affect that. And that's all.